Hello everyone, my name is Alyssa and today I'll be talking you through the second page of the biochemical tests for our class. This is part one of the exercise. So I'm basically going to be going over the urea slant, oxidase test, and the catalase test. So I printed this out. Sorry, it kind of has... my printer had trouble printing out this page. So the first thing we're going to do is we're going to sterilize our inoculating instrument. So in this case, for the urea test, we will actually be using a needle. So I'm going to sterilize that needle in the micro incinerator. And then what I'm going to do is I'm going to take, kind of make sure the source culture, the tube is open, and I'm going to sterilize the lid of that within my micro incinerator. Once that is sterilized and my needle is sterilized, I can take my needle, kind of dip it into the source culture, and then pull it out and then sterilize the lid of the source culture again before I put that lid on. Now that I have my inoculating needle, what I'm going to do is I'm going to take my urea slant. Let's just say, I guess this clip is my urea slant. So the first thing I'm going to do is I'm going to stab it about 3/4 of the way down to make sure it's fully inoculated, and then on the slant side I'm going to streak it up along the auger so that way we make sure it's fully inoculated. This test, what it's going to do is going to test for urea production. So basically we're going to incubate it for about 20 to 48 hours, I suppose, and if it turns a bright pink color, then we know that it's urea positive. So my next one is my catalase test that I'm going to go over. So basically what we're going to be using is we're going to be using a microscope slide. We have our source culture. So again, same process for opening that. But before we get into that, the first thing what we're going to do is we're going to go to our reagent cabinet. This is right over here, and we're going to pull out hydrogen peroxide. We're going to put kind of like a dot on the slide from our reagent cabinet. Then we'll go through the process of getting our source culture. So opening that lid. So opening the lid of our source culture with our thumb, sterilizing it in the micro incinerator. And then we're going to grab a stick from the cabinet and kind of just get a little bit of our organism, pull it out, sterilize the top of the lid with the micro incinerator, and put the cap back on. We're done with our source culture. Now that I have the stick with the organism and I have my microscope slide right here, what I'm going to do is I'm going to put it in the hydrogen peroxide. If it starts bubbling immediately, that means that it's catalase positive. And then once we're done with that, we're going to air dry and heat fix the slide, so make sure that there's no possible contamination as far as like any of our work surfaces. So heat fix, and then that's going to go like in a glass kind of any container in the lab for glass, and then we would put our sticks in the biohazard. The last test I'm going to go through is my oxidase test. So basically same process as we're obtaining our source culture from our slant, pulling off the cap off the tube, sterilizing the lid of it in our micro incinerator, and then what I'm going to do is I'm going to take a cotton swab. This cotton swab should remain sterile. So if this is the bottom of the swab and this is the top, I'm going to feel for the top through the packaging and then kind of poke it through so that this doesn't get touched by me. And then I'm going to pull it out of that packaging. And then we can use this later for disposal. So I'll take my swab. The lid is open and already sterilized from my micro incinerator. I'm going to dip it into my slant culture, pull it out, and then I'm going to sterilize the lid, or the top part, I suppose, of the source culture though, that I can put my lid back on. Okay, source culture is done. Now that I have the growth on my swab, what I'm going to do is I'm going to go to my reagent cabinet, which is over here, and grab the oxidase. So that if my growth is right here on the bottom of my swab, what I'm going to do is I'm going to gently put like 1 to 2 drops of oxidase on my growth. And if it turns purple, like a dark purple bluish color immediately, that means it's oxidase positive. If it is kind of like slowly changing color, we would consider this a negative. Once that's done, I'm able to put the swab back in that same sort of wrapping that I had before. Again, making sure not to touch this part that has my growth. So putting it back in the swab and then going into the garbage, or biohazard, excuse me. Yeah, and then once we're done, kind of like going through our especially after the urea slant, making sure we sterilize our work surfaces and also flame our needle to make sure that it can be used for any other tests that we may use. So again, just putting that in the micro incinerator and then kind of putting it to the side and sterilizing our work surfaces with a disinfectant. And yeah, that's it. Thanks for watching.